All right, hi everybody. Let's go through our review for chapter 9. This was dealing with microbial nutrition and growth. There were a lot of kind of random concepts in this chapter, which I didn't really much care for, but they are very important ones. So let's kind of talk about that. We talk about the important elements that are involved in cells and building cells and what's required to build a cell. So those elements and their associated macromolecules that we may be getting from our diet, or we could be getting them from organic sources or inorganic sources. So heterotrophs are organisms that get their carbon from organic molecules that they got from other organisms, by the way. Autotrophs, they get their carbon in inorganic form, usually in the form of carbon dioxide, especially when we are talking about plants. So that's the carbon usage. As far as the energy, where they get the energy from, phototrophs, these guys get energy from light, and then chemotrophs get their energy from chemicals, typically from breaking chemical bonds. It releases energy and they can store it in another form of energy. So, then we talked about the different combinations of those, which were like the important ones to me. I feel like the chemoheterotrophs, that's like us or any other organism that isn't going to be fixing their own carbon or relying on light for energy. So that's like us and a lot of the bacteria. And then we had the photoautotrophs, which are going to be using the energy from the sun often to allow them to fix the carbon that they're going to put in their carbon-based molecule into an organic form. So that's why we eat them, because they did that for us and now we eat them. And so anyways, the photoautotrophs, those are our primary producers because they did all that from scratch for us. So thanks, guys. Next we have all right, that was that. Okay, so then we have growth factors. So we talked about growth factors. These are things that have to be organic molecules that have to be provided to an organism for them to survive because they cannot make it themselves. So my example was like with people and vitamin C, how you have to have it provided to you in your diet. You cannot make it for yourself. Saprobes are organisms that digest dead things. So dead stuff, decaying stuff, that's called detritus or detritus, depending on how you want to pronounce that. But the saprobes break down the dead stuff. They can't necessarily like eat up or open up their cell walls to engulf something. So they secrete enzymes to break things down around them and then can absorb it from their environment. So halophiles, these guys flourish in salty habitats. Thermophiles, they like the hot temperatures. Psychrophiles, those were the cold ones, and the mesophiles were the medium temperatures. You do need to know both of those as well. They're just not on this. Next, we have the diffusion. Diffusion is movement of molecules from high to low concentration. We made all these comparison with bouncy balls in my office and bouncy balls in Dr. Rhodes' office. But anyways, diffusion is movement from high concentration to low concentration. A lot of membranes are semi-permeable or selectively permeable. Selectively permeable meaning that they will allow water to cross, but they won't allow most solutes like salt or something to cross. So so the water can move and equal out a concentration of something, but it cannot necessarily allow the other thing to move across. So the cell's really concerned about that. So it wants everything to be the same on both sides, inside and outside of the cell. And so when that is the case, that's called isotonic. Whenever there is more water outside than solute, so like let's say fresh, straight up water, that would be an example of hypotonic environment. And then in that case, we would be taking up the water to try to dilute everything that we have inside of our cells. And that would lead to bursting the cell potentially, overfilling it, though bacteria have cell walls that will help maintain their cell shape when this is going on. And then we had hypertonic, where that was the more salt outside, right? So we have halophiles, which are osmophiles, by the way, that happen to like salt. So they actually pack themselves with some salt in their cells so that when they're in their high salt environment, they're actually isotonic for them, but it wouldn't be to us. So that's how they deal with salt. But yeah, Otherwise, you can have a lot of osmotic pressure that can cause cell death in the way of plasmolysis. So yeah, that's what happens whenever there's too much salt or something outside. So that is hypertonic. Next, yeah, we talked about solute. So those are the molecules that are dissolved in typically the water. That's our solvent in a cell. So we did talk about this. Okay. Next, we have facilitated diffusion. This is helping molecules move with a gradient. So still high concentration to low concentration. We're just like creating a door for them to walk through. Okay, but you just have to have the pass to go through it. But we're still moving all people from one area where there's people packed into the area where there's not people packed. So that's still moving with the gradient. Active transport, however, requires energy. So it's not just going to happen on its own. So you're going against the gradient in this case, or you're just trying to move things faster than what diffusion would do. So that requires energy to do that. You're fighting what is natural. We talked about oxygen and gas requirements for organisms. We talked about aerobes that need oxygen. We talked about anaerobes, which do not need oxygen. They do not use oxygen. Facultative anaerobes are different though. They're kind of in the middle. They will use oxygen if it's there for aerobic respiration, which we'll learn about in the next chapter. But they use right. They use oxygen when it's available. I'm so sorry. Their facultative anaerobes will use oxygen when it is available. And then microaerophile, and if it isn't available, they undergo fermentation. So facultative anaerobes undergo fermentation in the absence of oxygen. In the presence of oxygen, they undergo aerobic respiration. Microaerophiles, these guys only require a little bit of oxygen. Aerotolerant anaerobes, these guys can handle the oxygen in their environment, but they don't need it. They don't need it. They'll just deal with it. And then we have obligate anaerobes. These guys will die in oxygen typically. They do not need an oxygen. They do not want an oxygen. Please don't put me in oxygen. I don't want the oxygen. So those are obligate anaerobes. They're obligated to be without oxygen, anaerobic. Okay, symbiosis, it's relationships between cells now, or organisms now. Symbiosis is whenever you have two organisms and their lives are intertwined somehow, and then one depends on it for survival. It has to happen, okay? It's required. That's symbiosis. Then we have commensalism. This is when one organism benefits and the other is unaffected. Mutualism, where both organisms are going to benefit from the relationship. And then parasitism, where one organism benefits and the other is harmed somehow. So that's the basic ideas for symbiosis. We talked a little bit about antagonism as well, because we were talking about microbial antagonism and how and abiosis. Those are basically whenever organisms don't depend on the relationship together for survival or anything. Neither of them does, but they sometimes will have a relationship with other organisms, like maybe just in passing or something. So we have microbial antagonism in our gut, where our gut bacteria might protect us from pathogens. Okay, that's microbial antagonism. Our gut bacteria do not require that. The bacteria passing through us pathogens do not require that relationship. So that is not symbiosis. So that's a non-symbiotic relationship. Yeah. So anyways, that's the only other one I guess I want to bring up with that. So as far as growing up, bacteria goes in cultures, we'll have a growth curve. That is what we use to depict the basic health of your bacterial culture and what stage it's in at any given time. And this differs from organism to organism. The lag phase is when the cells are beginning to be put into a new environment and adjust to it and start, replicating their DNA and adjusting and all that, getting ready to divide. Next, we have the log phase or the exponential growth phase, which is exactly what it sounds like. We're doubling and doubling and doubling and doubling over and over and over again. healthy, we've got all the resources we need. That could go on forever if we could keep it that way. The stationary phase is whenever we have a slowing or stopping of the growth rate. It usually means that we have growth equals death, or we're just not dividing anymore. So that's a flat level. And then we have death, which is going to eventually be exponential death rate. So loss of the cell or death of the cells due to, just, no more nutrients, or it could be, buildup of toxic byproducts, waste products, or something like that. So that's also a possibility. So that is that for chapter 9 review, and I will see you guys for the next one. Bye.